Hi students, today we are going to discuss about the cell organelle, important cell organelle. For any cell organelle question, either mitochondria, lysosome, Golgi apparatus, peroxisomes, endoplasmic reticulum, for any organelle, first you have to draw the cells diagram, right? One membrane, nucleus, cytoplasm, like that you have to first draw the diagram. Then proper labeling is must, okay? Then your question is lysosomes. So first you have to define what is lysosome. Second is what are all the functions of lysosome. Third is what are all the clinical disorders involved in this lysosomes. If there is any functional disruption, what are all the diseases they will go for, right? 3 points are very, very important points for our short notes, right? So lysosomes definition is they are organelles formed from Golgi apparatus and dispersed throughout the cytoplasm, right? They are formed from Golgi apparatus and dispersed throughout the cytoplasm. Second point is they contain some sacs, membrane bounded sacs, small vesicles like sacs, which contains hydrolytic enzymes. Very, very important, hydrolytic enzymes. Examples are proteases, lipases, nucleases and sulfatases. So this is the introduction point for lysosome. Second, what they are asking is what are all the functions? We have only two functions, right? So definitely you should write these two functions, otherwise they won't give marks. First one is these lysosomes involved in digestion of, this word is very, very important, digestion of intra or extracellular substances, that is foreign particles, that must be removed. Very simple, removal of the foreign particles, that is the role of lysosome. Second one is very important, another function, during development, that is in the embryo, it helps in the formation of fingers and toes. In between the webbed tissues, they only remove the webbed tissues in the toes and fingers. That is why they are useful in the formation of toes and fingers. Third, what they are asking is name the clinical conditions for this lysosomal disorders. First one is gout, hyperuricemia, right? Second one is silicosis. This is one kind of respiratory problem. Third one is eye cell disease. This is lysosomal storage disease. Another name of the title is lysosomal storage disease. The examples are Tay-Sachs disease, Pompey's disease, like that. So you pick up any 2 or 3 for your clinical conditions related to lysosomes. Okay. Next important cell organelle is peroxisomes. This is also very, very clinically important cell organelles. So what is the definition? It is also membranous sacs which containing enzymes. These two are very, very important MCQs. Catalase and peroxidase. Catalase and peroxidase. Perox, hydrogen peroxide, removal of hydrogen peroxide. So it is a membranous sacs. These peroxisomes are membranous sacs which contain the catalase and peroxidase. Very, very important MCQ enzymes. Functions. They involved in the process of xenobiotics or detoxification. This is nothing but removal of the toxic compounds should be converted into non-toxic compounds and get excreted by kidneys. That is the definition for xenobiotics or detoxification. Second one is they are useful for the synthesis of some phospholipids, example glycolipid and plasmalogens. So synthesis of phospholipids. Next one is what happens in peroxidation of PUFA. PUFA means polyunsaturated fatty acid. Polyunsaturated fatty acid peroxidation. They will release some free radicals. FR means free radicals. You know, free radicals are very harmful to body. So they damages the cell membrane, tissues and sometimes genes also. So for this free radical, the scavenger is this peroxisomal enzyme, catalase and peroxidase. So what is the role of peroxidase and catalase? Removal of the free radicals, right, occurs by peroxidation of polyunsaturated fatty acids. So these three are the functions of peroxisomes. Coming to the clinical diseases, we have so many diseases, but these two are very, very important diseases. Zellweger syndrome. Another one is ALD, that is adrenoleukodystrophy, right? Zellweger syndrome, what happens? There is a defective formation of peroxisomes that leads to accumulation of saturated long chain fatty acid. S means saturated, LC means long chain, FA means fatty acid. In liver and brain, the accumulation of defective peroxisomes, right? So what happens is they will go for neurological symptoms and sometimes death also. That is why this is very, very important clinical disorder involved in peroxisome Zellweger syndrome. Second one is ALD. This is also the defect in the membrane protein of the peroxisomes. Some mutation happens in the peroxisomal membrane that leads to adrenoleukodystrophy. This is also very serious condition for the, right?